Hello guys, welcome to this video. In this presentation, we present how to clone a selected gene into E.coil cells step by step. In our presentation, we will try to clone hepatitis E virus or HEV gene into E. coli and get them express virus-like particle for HEV vaccine development. Our work is mainly inspired by this article in which they try to express HEV virus-like particle from E. coli cells. To clone gene into E. coli, it can be divided into two steps. Part A, plasmid vector preparation, copy selected gene into a carrier. Part B, bacterial transformation, inject the plasmid vector into bacterial cells. The first step is to extract the genotype of HEV virus. According to the article, HEV samples are collected from HEV infected pigs. Samples are tested with antibody diagnostic kit to ensure infection. The only positive test samples are extracted by reverse transcription and gel electrophoresis to obtain HEV gene. Then they are purified and analyzed whether or not they are HEV genotypes. Next, we clone only specific genes of the HEV virus by PCR method. The genes that express virus-like particles are chosen by the primers, which bind in the E1 and EcoR1 sites. The PCR parameter is 30 cycles. At the end of this step, we now have numbers of genes to transfer into E. coli cell. But we need to send them as a plasmid vector. So we need to prepare the plasmid first. This step is not mentioned in the article, but in general, there are two methods to isolate plasmid. First, alkali lysis. We put E. coli cell in the alkali medium and set the cell will break. However, DNA structures are denatured into single strand due to basic environment. Acid needs to be added to neutralize the solution and renature the DNA structure. To purify, we use centrifugation and alcohol precipitation to obtain the plasmid. Second way to isolate plasmid is boiling lysis. The purification process is mostly the same, but the cell lysis is caused by the heat of boiling water. This method is much faster but less yield because high temperatures can degrade DNA into fragments. Now it is time to insert HEV gene into plasmid. According to the article, PMD18T plasmid are used as vectors due to compatibility. The plasmid are digested at NDE1 and ECO1 site by restriction enzyme to open the circular structure. The HEV gene is inserted automatically due to compatible site and connected by ligase enzyme. At the end of this step, the plasmid vectors are ready to transfer into E. coli cells.
The next step is transformation, which is the introducing sulfur and DNA in form of plasmid vector containing the genes of interest, which in this case, the HEV gene into bacteria like E. coli. A step start with the preparations of E. coli cell, which going to make the E. coli cell competent. It's normally done by treating the cell with a divalent cation, for example, calcium ions, to make the cell membrane more permeable for the plasmid vector to get inside the cell. Next is storing the competent cells at low temperature, then adding the HEV DNA. After that, the mixture is suspected to heat shock at high temperature, usually around 42 Celsius for a short period, to create temporary pores in the membrane for the DNA to enter. Then return the cell into ice immediately for stabilizing the E. coli cell membranes and reduce the chance of the cell taking up their own DNA. After that, transformed cells are allowed to recover by placing the tube at moderate temperature for a short period for repairing the damage caused by the heat shock and start expressing the gene from the newly introduced plasmid. Next is the selection step, which is the identifying and isolating all the E. coli cells that have successfully taken up the recombinant HEV DNA. In general, plasmid vector also contain antibiotic-resistant gene along with the HEV gene. So, to check for the transformation, we grow those cells on a plate coated with kanamycin antibiotic. Only survived bacterial colonies are cells that successfully taken up recombinant plasmid and should contain HEV virus-like particle gene. After incubation, surviving colonies are visible on the agar plate. The next one is screening step. It is about analyzing to confirm and characterize the presence of the desired gene in the transformed bacteria cell. Individual colonies are picked from the agar plate, which are the colonies that carry the plasmid with HUV genes and antibiotic resistance. Next, the DNA from each selected colony is extracted. This can be done by using alkaline lysis or commercial DNA extraction kits. The numbers of extracted genes are multiplied by PCR method. To select only desired region, the same primers are used for PCR. The primers bind at the NDE1 and E.coil site. Following that, the PCR products are run on the agarose cell electrophoresis and separate the DNA fragment based on their size. The gel then visualizes and the presence of expected DNA band confirm that the genes of interest is present in the selected colonies. After knowing which colonies contain HB gene, next is expression step. In cloning, it involves inducing the transcription and translation of the clone gene within the host cells to produce corresponding protein. First, the plasmid used for cloning typically contains promoter region that regulate the initiation of transcription. Upon induction, this promoter become active. The inductions of the expressions of the clone genes can be done by adding a specific inducer to growth medium. Commonly, inducers are IPG. Then the activated promoter leads to the transcription of the clone gene into mRNA. The mRNA carries the genetic information from the gene to the ribosome. The ribosome then read the mRNA sequence and synthesize the corresponding protein by linking together the amino acid. The newly synthesized protein undergoes folding into its functional 3D structure. In some cases, post-translational modification may occur. And the last step, which is the harvesting step, it is about collections of the bacterial cell. After inducing the expressions of the clone gene, the bacterial culture is allowed to grow under conditions that promote the synthesis of the desired protein. Following that, the monitoring growth the bacterial culture to ensure that a significant amount of the protein has been produced. Then the optimal harvesting time is selected. Harvesting too early may result in low protein yield, while harvesting too late could lead to degradation of the protein. Following that, the periodic samples are taken from the culture to assess the expressions of the target protein, the presence of virus-like particles, and assess their expression level can be confirmed using SDH page. Once optimal expression period is reached, the bacterial culture is usually centrifuged to form a pellet to sediment the cell at the bottom of the centrifuge tube. Then after harvesting, the bacterial cell need to be disrupted to release the intracellular components, including the synthesized virus-like particles. Following that, the disrupt cell lysis is suspected to clarification to remove insoluble cellular debris by centrifugation or filtration, separating the soluble fraction containing the virus-like particle from the insoluble component. The superminant or liquid portion above the cell pallet is carefully removed to prevent contaminations of the unwanted media. And the last step is analyze the properties of HEV virus-like particle. Many characteristics can be observed, such as the distributions of the particle in the solution by the DSL or dynamic light scattering. The shape and the size can be observed by using the TEM, and homogeneity of the protein can be observed using the atomic force microscopy or AFM. Thank you for listening.